Okay, good afternoon everyone, and welcome to the Waite Research Institute Seminar series. This week we have Associate Professor David Jeffery speaking on the synthetic and analytical chemistries enabling grape and wine science.
Thank you very much, Rebecca. And I'm going to use the mouse on here for the audience that's joining us at home. So I'll try and, focus my attention on the screen up here for the audience that's here and the one at home. I chose this lovely background here because if you might notice someone standing in the background there, which is me from a couple of years ago. So I thought, why not promote myself in multiple ways, hey? And when the down arrow comes alive, and it doesn't want to. Here we go. I thought I'd start off with, it's like a story, a little bit of a story. So a bit of background, who am I basically, and how did I end up here? It's a great question we probably all ask ourselves. So I started off Flinders University graduate, Bachelor of Technology, Forensic and Analytical Chemistry, and then went on to do a Bachelor of Science and then a PhD in Synthetic Organic Chemistry, and that's what I'll touch a little bit on as I go through this talk. So what that did is gave me a good background in organic and analytical chemistries, which I now then use to as tools of the trade that, with the projects that I work on these days. So giving me very good synthetic capabilities, but that ordinarily comes along with doing analytical chemistry, also comes along with doing a synthetic project because you're doing lots of analyses to characterize compounds. So I'll show you in a second. I'll run you through every step of my total synthesis that I did for my PhD and see how quickly you fall asleep. But I made this molecule here that was extracted out of this lovely marine sponge there that the biologist went and ground up and got its guts out and ran it on a TLC plate and identified UV active compounds and determined this structure. But as it turns out, they got it wrong, and that's what my PhD kind of showed. So in terms of my career, I'll go into that synthesis in a minute, every single detail and every step I'm sure you care about. But in terms of now my career afterwards, I started out at the Australian Wine Research Institute and worked there for about 5.5 years, working my way up the ladder there, which was I knew nothing about wine beforehand. So that was an awesome introduction to the world of wine and wine chemistry, but they didn't care. They wanted someone who knew chemistry and analysis and understood that. And I guess they figured they could, I could learn the wine stuff along the way. And indeed I did. And after starting at the university and working my way up and now stagnating as associate professor, I wrote this textbook a few years ago with a couple of guys from one in Davis in the US and one at Cornell University. And this is kind of one of the highlights of my career, I think, writing this textbook that we now make every student buy that I teach to. No, of course we don't, because we provide it through the library and they can download every chapter and put it up on another server somewhere and then other people can download it. We try not to promote that, but so yeah, a couple of things along the way. I've done a graduate diploma in online learning here at the university and got a couple of awards. This book actually received a special mention from the OIV in terms of an enology textbook at the time, a couple of years ago now. And we've put it off for a while. We figured after 5 years we'd revisit this. I thought, I'll never write another whole textbook again. Definitely not. It was a lot of work. But yeah, we might do a revised edition where they're looking for about 10% of that content to be updated. So let's hope we can do that quicker than the 3.5 years it took us to write the original one. So this is the stuff I want to scare you with. And I'm not going to dwell on this, but I just wanted to show you, give you an appreciation for what does a total synthesis look like. So basically it's like molecular origami or Meccano or a micro scale, things that you can't see, and you're putting these molecules together and then you go and do the analysis and prove what they are. So there's just a couple of things really I want to highlight, the challenges. In terms of starting out, you can work on very big scales, so 15 grams of this thing I converted into this thing, but you're always losing yield along the way. If your steps aren't highly efficient, then if you get a 90% yield and then you get another 90% yield, all of a sudden in 2 steps you're down to 81%. And if you keep that going, when you're trying to take material from a starting point and take it all the way through to the end, you can end up with bugger all of the compound you're interested in. I'll highlight that as we go along, as well as discovering some interesting chemistry in that. So that's kind of the point of total syntheses. That's the new molecule, or the verification of the structure and the stereochemistry that someone else has ballsed up potentially, but also developing new synthetic methodology, which is what came out of my project as well. So, and you do cool things like get a crystal structure. So you work with those sorts of people as well. And sometimes crystal structures alone can be a publication, which is kind of neat. And so taking that a bit further, I had, so back to 14, that crystal structure, I had 100 mgs of that, and I took it through to this step. And there's lots of stereochemistry in that involved. I'm not really going to get into that. Here's a novel step here, this like a dehydration step. But look, now I'm down to 20 mgs. And then I put a methoxy group on here, and suddenly I've got 10 mgs and 8 mgs of this gear. So not much to play around with at all. And you've got to do all your analyses on that as well, you know, so HPLC or UV-Vis or NMR. So yeah, you're really mucking around with very small amounts. But you know, that's what we do here in the lab as well, in terms of we start out trialing a reaction maybe on 10 or 20 or 50 milligrams, not a lot of material. And typically we've got oodles to play with here. You know, we're not talking about 20-step synthesis like I did for my PhD. So I kind of alluded to this before in terms of my first job was at the Australian Wine Research Institute. They weren't so worried that I didn't have a wine background, although they asked me what sort of wine I liked, and I gave the shittiest answer ever. Like, it was just something I could think of at the time. Because yeah, I wasn't a huge wine drinker, and probably looking at my belly, you can tell that I prefer beer still. So yeah, my background was very helpful in getting that position, and I'm very grateful that that was my start here in Adelaide. So I wanted to hang around in Adelaide for family reasons and that, and you might realise there's not a lot of places where you can go and work in Adelaide in terms of doing research or whatever. So I was very grateful to have that, to obtain that position there at the AWRI. And similarly, I was really grateful when Graham Jones decided to retire, because that meant I could just, well, I applied for that job and got his position. And, you know, if you know anything about academia, you either wait for people to retire or croak it before a position becomes available. And it's hopefully the first one, of course, you know. So, yeah, I got the position here at the university in 2010. And instead of walking to the 3rd level of the building up the road, I walked to the 4th level. So presumably my next position will be on the roof, I don't know. But yeah, we're not really allowed to go up there, I don't think. So why wine, or why is that of an interest? You know, I'm kind of preaching to the somewhat converted here in this audience, but it's a multi-billion-dollar contributor to the Australian economy. It's a big employer. This last vintage, about 1.5 million tonnes of grapes were crushed, and there's a levy associated with that. And on the international stage, you know, we're a pretty big player, relatively speaking. So 6th largest producer, and it's hidden behind the bloody video there, but I think it says 5th largest. I think we're the 5th largest exporter. Is that right, Lyra? Yeah. And thousands of jobs, and these are regional as well. So that's what's really important, right? You know, it's not just people sitting in offices in the middle of town. It's actual, these things are important to the regional economy. I've shown you, you always got to have graphs. So there's a couple of graphs there, but no, this is around tonnes crushed of red grape varieties and white. So you might know Shiraz is our most crushed red, followed by Cabernet Sauvignon. And in terms of white, it's Chardonnay, and I don't know, somewhat surprisingly, Sauvignon Blanc is there, and a couple other white varieties, but Chardonnay and Shiraz are our big ones. So analysis. Analysis is the key to the success, or at least the success of what we do. So there's an old adage, and it's not mine, but you can't, and this applies to business and everything, you can't manage what you can't measure. So how do we measure things? That involves analytical chemistry. And in fact, analytical chemistry, or chemistry in general, has been very important to Australia right back in the 30s. And something as simple, or we think it's simple nowadays, something as simple as pH control, you know, really revolutionised the industry. And, you know, back then, the gentleman had to argue with the manager to buy a pH meter, you know, to measure pH. But, you know, this really turned the industry around in terms of understanding pH and acidity and spoilage and that, which was really important at the time. But what we're talking about in general, I mean, that's a really basic thing. We still measure pH today, and it's still important today. But what we're really interested in is all the other things that sit underneath that, you know, the minor components basically. So things like polyphenols, sugars are a bit more of a major thing, amino acids, the aroma compounds in particular, and their precursors from the grapes or wherever they might come from. So there's plenty of things there. The other thing to think about is lots of different chemistries, and So different functional groups, different reactivity, different concentrations. So this often requires multiple approaches, analytical approaches. If you want to measure 100 aroma compounds, you probably need 5 methods to do that. And if you only use 5, or if you only need 5, that's a bonus. So it just kind of highlights the scale of the issue in terms of they're variable in concentration and different in reactivity and respond differently to different instruments. So often you need a range of analytical methods to be able to do this, and that's what we do. We develop those methods, but not just for the sake of it, we want to apply them as well. So the types of things that I've listed there and that we're interested in relate to sensory properties, of course, and then therefore how do consumers enjoy wine like that, but also things around quality and style, and there's faults that come up occasionally that need to be solved. And but a lot of this just is around understanding the product, what feeds into that, what creates that product, the underlying composition, and then how does that relate to what consumers want to consume, because ultimately we could understand everything about wine, and if people aren't buying that wine, then it's not much good for the industry or that particular business. So not only do we deal with reactive things of different concentrations of that, they come from multiple sources. So this is really interesting, and you probably know around the campus there's people that work in certain areas, so microorganisms, smoke taint, for instance, with Kerry's work, things around barrel maturation. So at each of these points here, there's inputs from the grapes during fermentation, of course, and even beyond that. So once you put it into package and the different closures, and then aging continues after that. So wine is an amazing natural product, and that's what kind of draws me or keeps me here doing what I'm doing, because it's never the same. It's constantly evolving, and to understand that, requires some real interesting or unique approaches to be able to understand these phenomena. So I'm only going to focus on a couple of these things related to what comes from, well, they originate in the grapes, and I'll explain that a little bit, but some flavour compounds in the wine, and some of this has been an ongoing story, and Dimmy's here, and we've been working on some of these things for years around these varietal thiols. So, some of our first work was easily a decade ago or more, and this is something that I still try to focus on and maintain a focus in this area, and have PhD, usually at least one PhD student working on this sort of thing at any one time. So I'm talking about varietal thiols, or also known as polyfunctional thiols. So they have more than one functional group. A thiol has an SH group. Polyfunctional, it has either a carbonyl or an alcohol or something else in there with it. So we can see here, and let me move this vid so you can see the cat peeing on the toilet. And yeah, here we go. So some of these compounds, or some of the main ones, and they're odor impact compounds. So they impart, they're very powerful or potent odorants. So you can see here 90 nanograms per litre, 90 billionths of a gram per litre for what we call MSP, the cat pee compound, or it smells like box hedge or black currant or other things. So these are incredibly low concentrations that you find them at, but also their thresholds as well. So you can see down here in the nanogram or tens of nanogram per litre range, the thresholds of these compounds. There's a couple of other things to consider. So thiols are very reactive, which makes them a challenge to analyse. They've got tending to have nice aroma characteristics. They're usually present at concentrations well above their threshold, so they're going to be important in terms of wine aroma. And for all those people that read in structures, you can see here that there's different stereochemistry as well. So that introduces another level of complexity into the analysis or understanding of these things, because our nose is a stereochemist, or our senses are a stereochemist. They can recognize molecules that have certain three-dimensional shapes, and often natural molecules are of a certain handedness or chirality, but also they can be, at least if it's chemically formed, then they can be present there in kind of equal quantities. But typically, one will usually exceed the other, and this might indicate the pathways to formation of these things. So I won't talk about the whole other area where these form from precursors in the grapes that are extracted. So non-volatile things, how kooky is that, right? Things that don't smell, and this is a lot of things in grapes, they don't have an aroma. And then after fermentation, suddenly they're released and we can perceive them. So that's kind of interesting. So what have we done in this area? Well, we develop analytical methods that then we apply to certain challenges or to understand what's going on in terms of in biochemical or chemical space, how these things evolve during winemaking and aging and that. So what we do is apply something called stable isotope dilution assay, which involves a stable isotope. So we need to be able to make those. You can buy plenty these days, much more than you could before, but they're quite expensive. But there's still lots of things that you can't buy. Or if it's easier for us to make it than buy it and much cheaper, then that's what we'll do. And similarly with the authentic compounds. So you can see here lots of deuterated positions in our standards here, and it doesn't really matter about the detail, but I just have a look at all the Ds. And what it means by stable, they need to be in a position where they're not exchangeable. So they can't sit next to a carbonyl. They can't be an acidic proton because that will just exchange in an aqueous environment. So that's what makes them stable. And so we'll produce these and also make the authentic compounds if we need to. And then we have our standards to be able to develop our method. And our initial go at this method looks something like this. And this is Dimmy's efforts from back in the day during her PhD in that, creating this method here. So I've mentioned around thiols, or designated as RSH. They're reactive, very difficult to analyse. They stick to metals, they oxidise, they do all sorts of things. And they're at very low concentration, so that requires really sensitive methods. Ain't nobody got time for working with mercury. That's this thing here, and it's probably a relatively safe form of mercury, but we wanted to eliminate or come up with a procedure where we didn't use anything to do with mercury. It involved derivatization of the thiol, so it's no longer reactive and it performs better in the instrument. We can detect it and analyze it much better if it's derivatized. It's no longer so reactive, but the sensitivity is enhanced as well. So this was Dimmy's effort, the separation flask Christmas trees, basically. So this is a whole series of samples being done with this method. We only did 3SH, the passion fruit kind of thiol originally, and then we intended to expand that, but in fact we went down another path. But using this kind of approach with just some liquid extraction and derivatization with this benzylating agent here, which puts lots of fluorines on, Dimmy was able to do like 25 samples a day. And that's not huge throughput, right? It's probably enough in terms of a research project and being able to understand things, but it's not massive throughput. The detection limit was great, but we were only doing one thiol there. So I thought, F that, there's got to be a better way. So I went and had a look at the literature, and there are different ways to derivatize things. And Renata helped do some of this work, actually first began this work with just a tiny little seed grant from what was Wine 2030 at the time. And we kind of flipped this around. So 3SH, or these thiols, they're volatiles, they're aroma compounds, they're in the gas phase. You analyse them in the gas phase usually. But I thought, well, let's be a bit kooky, and wine's a liquid. What can we do to this so we don't have to extract the volatiles? Let's analyse them in the liquid phase, and that's exactly what we did. So I went and found out there's a couple of different derivatizing reagents you could use, but here's our thiol. It reacts with this dithiodipyridine reagent, so via thiol disulfide exchange, basically swapping around what that thiol was joined to. And we get our derivatized thiol here, and then this thiopyridine. That doesn't matter so much, but this can be, as you see there, there's some lambda maxes written. This derivatization procedure is amenable to UV-vis spectroscopy, for instance, but we're talking about thiols at really low concentrations. So we did a bit more than just derivatizing the thiol and analysing it straight away. We had to have a concentration step. So this is the procedure, 20 mils of wine, which is a lot compared to what you might analyse other things. You might only need 1 or a couple of mils usually of a sample, but we need this concentration factor because of the low level of these thiols. So internal standard, labelled internal standard, a mixture of the compounds we're analysing, EDTA to mop up any metals that might be hanging around and want to do nasty things to our thiol. Acetaldehyde, this was an interesting step, which actually tied us up for a little bit, but Renata had to think about it, and it's obvious now, but in other analyses of sulfur compounds, you add acetaldehyde to bind up free SO2. If we didn't do that, or when we didn't do that, this thiol derivatizing reagent would just be completely consumed by the SO2 in wine. So this was a neat little step, adding that to bind up any free SO2, then adding the derivatizing reagent, putting it through SPE to be added, basically a little bit of a cleanup and concentrate down by 100-fold. Trap for the new players, make sure your SPE cartridges have sorbent in them or they're not going to do very much. Drying those samples down, reconstituting and analysing by HPLC with tandem mass spectrometry. And a particular method called multiple reaction monitoring, which I'll show you some chromatograms in a minute so you can see how clean that is when we do this. We were also interested at the time, there's other thiols, so some of these I mentioned on the earlier slide, 4-MMP, 3MH, 3MHA, or they go by their kind of modern names as well, 4-MSP, 3SH, 3SHA, but there's also benzyl mercaptan and furfuryl another couple of thiols that are really potent compounds. So roasted coffee for furfuryl thiol with a below 1 nanogram per litre threshold, that's incredibly intense. More powerful compound. And then this benzyl mercaptan, it's called empyreumatic, can hardly even say the word, but it basically means smoky. We needed internal standards for those. So what did we do? We went and made them. And this is pretty straightforward sort of stuff here, chemical-wise, in terms of how complex it can be. So, and in a way, we don't care about yield so much when we make these, whereas I did when I did my PhD, but we're not taking these through multiple steps. And in the end, because the thiols are in such low concentrations, we don't need massive amounts of these standards anyway. So if we got a 20% yield and made 50 mgs of that, that'll last us 100 years. This is what I'm talking about in terms of doing a multiple reaction monitoring method now. So we validated this method and there's certain steps to go through. It was important that the limits of quantification around the aroma detection thresholds of these compounds, so small changes that may affect aroma, we can detect them with our method, which is great. And then the tandem mass spectrometry means we can do multiple reaction monitoring. So basically, we look at how these compounds fragment in the mass spec. So there's the derivative of 3SHA and there's its internal standard. And you notice, so the internal standard, this deuterated standard, behaves exactly like the natural analyte. If that's going to stick to something, then so will the internal standard. If you spill this gear, then a proportional amount of internal standard is lost, or if you concentrate it differently. So that's the beauty of these deuterated internal standards. Their only difference is the molecular weight, or the M/Z that you get for these. So you can see that's 5 mass units different. Often that will carry through in the fragments as well, but it depends how this molecule fragments. So it falls apart at this bond here, and that leaves that 144. And when that fragments, we get down to a 111 or an 83 or whatever. So from this fragmentation pattern in the mass spec, we choose those ions that we want to record, and we only care about those ions. All the rest, whatever other molecules are in there that don't have these ions, the instrument doesn't even see them. And therefore, we get really nice chromatograms like this. And these are real wine samples with thiols at the levels that you would find in wine. So, in the nanogram per litre range, or several hundred nanogram per litre range in the case of 3SH. But you can see there's absolutely no problem with picking out the peak of interest. And you can see there's hardly any noise. And that's the beauty of tandem mass spec and this MRM method. So this is another highlight of my career, Analytical Chemistry, this journal there. Right? So yes, I'm a analytical chemist / synthetic organic chemist masquerading as a wine scientist. So I still like to publish in really good journals in the field of analytical chemistry, but of course I code them during ERA to 0706 MAT. So where do we take this now? I was mentioning chiral molecules. So we want to understand stereochemistry. How does this impact aroma? What's going on biochemically? The yeast spit out certain ratios of these things. So we needed to develop a method. We took that liquid chromatography method with the TDP derivatization that we had worked on and converted that into a chiral method. That involves screening different columns. You don't just go out and buy a chiral LC column every day of the week. They're like $2,500. So we had to make this authentic derivative, which we don't normally do. We just take a sample, that 20 mils, and spike things and then analyze that. And you're talking, barely femtograms of, or, maybe attograms or something of compound in that you actually inject into the instrument. Whereas doing chiral column screening, so we didn't have to go and buy heaps of $2,500 columns, we produced the authentic derivative, which we never do, and we don't normally isolate this, but using a bit of simple chemistry, we made the authentic derivative. So we could send phenologics, they're called, did chiral column screening. It's basically Phenomenex, and it's a service they offer for free because obviously they want to flog you a $2,500 column at the end of it. So we had to send like, and they don't do mass spec, so it was UV vis, so we had to use high concentrations of this gear. We had to send, you know, tens of milligrams of this stuff or make a solution that contained a strong enough amount of this compound for them to detect it just using a UV vis detector. Anyway, they gave us some options on columns, and we tried the first one, and it didn't give us the resolution that they said. It was close to resolving the enantiomers, these top ones, but it didn't resolve them completely, whereas it did for 3SHA. Anyway, we went back to them. So we bought our $2,500 column and we tried it, and we did a couple of months. Our student worked on this for a couple of months trying to figure out what the issue was, but it was just the column in the end, or the, you know, it wasn't the appropriate phase. There was another option, which they ended up sending us for free, you know, so good on them, because we bought the column they suggested and it didn't work. But so we can see this nice separation of the 3SHA enantiomers, and this crazy separation, that is very rare, or you hardly ever see separation of enantiomers by that much, like 10 minutes or so, or 20 minutes in retention time, which still weirds me out till today, but I don't dwell on it. We just go ahead and do what we need to do. Again, we can collect the fragmentation data or the certain ions of these compounds. So that's what the mass specs look like. And basically, it's another MRM method. You really don't see much else in here other than the analytes of interest, and hiding underneath those are the deuterated standards as well. So when developing a method is not the end of the road, it's what you actually do with it and what knowledge you create, or what problems you understand or create also, maybe. So we applied this to a range of commercial wines and had a look at the different enantiomer ratios, and you can see some of those here from one of the particular studies. So another great analytical chemistry journal here that Liang published in, which was his first paper, I think, which is pretty amazing. So we looked at enantiomer ratios, and they were roughly 50-50, which is known between R and S, 3SHA, the two enantiomers, but a little different when you get to 3SHA. Did I say 3SHA? Anyway, 50-50 roughly for 3SH, and a little bit different for 3SHA. And this likely relates to formation pathways and the chemistry involved as well. So things, even if they're naturally produced at a certain or very enriched in one enantiomer, then chemically some things, and I don't believe it's these, but some things can rearrange and form the other enantiomer till ultimately they equilibrate to a 50-50 mixture. But the difference in enantiomer ratios implies something here about formation pathways. And then it jumps up even more when we talk about botrytized wine. So a fungal infection on the grapes, that really alters the enantiomer ratio as well. So there's definitely something enzymatic going on there. And I'm not a biochemist, so we just sort of dabble around the edges of this stuff. But yeah, we're still identifying and some, you know, interesting information, and other people go and take this and build on it, and then we take it back and build on it again, etc. So we have now linked this stuff, and I don't talk about the precursors at all. That's a whole another 10 years of work there. But now we can link the stereochemistry of precursors to the stereochemistry of the free thiols to understand, again, around formation pathways of those precursors, how they're converted to thiols, and then what happens after that. We're interested in the fate of thiols as well. So what a great segue that I remember this slide was coming up. So chirality and the fate of thiols. So being a chemist, I'm always thinking about chemistry things, and I thought one day, and it was weird, Liang, my PhD student, I just wrote this on a post-it note. I said, you need to look for this compound. And as it turns out, it's there, and for some reason the text has gone a bit wacky here, but so 3SH, it's a 1,3-alkanol, this special relationship here, and there's acetaldehyde in wine. It's either formed during fermentation or during oxidation through storage, oxidation of ethanol. So I thought, under acidic wine conditions, these things should surely be able to react together. I know that acetaldehyde, or not just me, everyone knows that acetaldehyde is an electrophiler and reacts with other things. It links tannins together, it does all kinds of things. And if they're both present in wine, then why wouldn't this thing form? It's in passion fruit. So it's good that it's somewhere else in nature, otherwise you start to wonder what's going on. So if you ever look at a paper and it's got all these weird methyl benzenes and stuff, you know, this is junk, it's not real things. So if you ever look at something and wonder about it, you want to know what's the natural formation pathway of those. And if it doesn't exist in another fruit or plant or something, then yeah, you might be in trouble there in terms of trying to understand. And it's very rare also to identify new compounds in wine these days anyway. So as it turns out, there's enantiomers of these things. So they come from 3SH from R, this series here, and this series comes from S. And there's also cis and trans isomers as well, which I won't go into, but it turns out that the natural or the more favoured form is the cis form of these things, and that was shown in the literature as well. So this is a new series of compounds we want to look at. We need a method, we need the standards to do that. So we produce those, deuterated standard. We could buy the oxythyne itself. It's a mixture of 85 to 15 of that cis to trans kind of thing. But anyway, we wanted to now show that that existed in wine. So we went through a few steps, comparing mass spectra, retention indices on 2 phases, co-injections of the analyte into a natural or a matrix containing this natural analyte as well to prove all these things. We produced the deuterated standards and quantified a series of wines then. So you might see a pattern here. It's like, hey, well, you need an idea first, then you either go looking for it or produce the standards, then go and identify it and go through all those steps, then develop and validate the method, then apply that method. And that's really the end game almost, is getting to the application of the method. And, you know, once we've got these methods from there on, we can apply them however we see fit. In this work, the odour detection threshold was determined, and the characters of this compound were already known. You can see here 7 micrograms per litre. That's like 1,000 times higher in threshold than the sulfur compounds themselves. So it's not likely that this is going to be an important odorant, but the fact that it gobbles up a certain proportion of 3SH, which is an important odorant, is where the significance is. So a series of wines were analysed and looked at the ranges of these things. They're only tiny, not very big, but it can consume roughly the same amount of 3SH. So 460 nanograms per litre was the highest amount here. We can see in Sauvignon Blanc, which Sauvignon Blanc is known to possess high amounts of those varietal thiols. Dim's done some work on Chardonnay, which is also appearing like it can have plenty of thiols in there, even though they're not known to be tropical styles. And then there's regional influence as well. So New Zealand Sauvignon Blancs, again, they're known generally to, some of them at least, to have really high thiol levels. So it's no surprise there that the oxythyne was present at the highest concentration. Australia, much less, and then France less again as well. So all this made sense in terms of what you might expect. So it's never the end. We're always trying to do more. So then we had a look at, Jing Chen, a new student has started or doing some work on this and taking over from Liang where he left off. So it's around the stability of these compounds, how do they form during fermentation. So I said before, thiols get released during fermentation, acetaldehyde gets produced. Does the twain meet? Well, it turns out they do. And so we had a look, a couple of different yeast strains. Vin13 is a thiol releaser. So from the non-odorous precursor release that thiol. And J7 is purportedly at least an acetaldehyde producer. So it's a sherry floor yeast. So it's not usually used to ferment grape juice, but we did because there is some data that shows it can still produce acetaldehyde during the primary ferment. So that's what we wanted, an acetaldehyde producer. It didn't turn out to be that, but that's not a big deal. Vin13 produced enough acetaldehyde. You can see here the acetaldehyde curves during fermentation for those different, for those different strains, which matched up very nicely with the oxythyne that was forming. So it seems that acetaldehyde, even though it's in much higher concentrations than the thiols themselves, that seems to be the limiting factor. And I better hurry it up a little because I've got a bit more to talk about. But so correlations down there, you can see with the different yeasts, the correlation between acetaldehyde and the oxythyne is very strong. Funnily enough, the correlation was quite weak for 3SH, but negative, which makes sense. So taking this further, if ASH, a 1,3-alkanol sulfonyl alcohol can form this thing. What about others that have that particular arrangement of atoms? So here's those ones I showed you before, but here's another sulfur compound which is very potent and has characteristics around citrus and stuff like that. So that also has a particular arrangement. So in theory, that also should be able to form or react with acetaldehyde and form a new oxythyne. And Jing Chen has just gone and done all that work. And I haven't, how rude of me, I haven't even, so I copied and pasted the references and just updated the dates and stuff. I haven't even changed your surname. So, well, sorry, Jing Chen. I'll make sure I do that afterwards. So we'll have to re-record all of this. So identified another oxythyne. How good is that? It is good because it gets a paper and that gets PhDs and it's interesting, really cool stuff. Like I said before, you very rarely identify new aroma compounds in wine these days. And there's about, 1,000 known ones. So now we're getting down to, identifying weirdo ones like this. So synthesize, you've got the idea, synthesize standards, develop a method, do the odor detection threshold of this. And we also proved that at least partially this forms under chemical conditions as we had proposed earlier, that acetaldehyde and these thiols come together in the acidic wine matrix and form this new compound. Whether there's any enzymatic involvement is another story. So in passion fruit, the enantiomers are very much skewed to one way than another, whereas we find them are much more even. So whether there's enzymatic overlaid with chemical is an interesting thing, and that opens up more questions that we need to follow up. But I want to talk a little bit briefly about authenticity because it kind of piggybacks on what we heard a little bit about last week. So wine fraud globally is a multi-billion-dollar issue. Even in Australia, a couple of years ago, the figures were like 300 million lost to the industry just in 1 year. You might have seen a film called Sour Grapes. You might have gone to your favourite store and picked up a bottle of Ben Folds. Probably not, because you wouldn't buy it here, but anyway. So there's different options to prevent this sort of thing from happening around blockchain, so track and trace and following things through the system and smart packaging and holographics and everything you can think of. But, those things are probably prone to being fraudulently reproduced anyway, right? Whereas we think that the chemical underlying chemistry, the molecules in there, and you could apply this to DNA and other, there's other things that are used for authentication, but I'm a bloody chemist, right? So what method will we use? Well, there's different ones. And we decided, Richira, my PhD student, looked at a range of different methods, and we were thinking about rapid ones that could be deployable within the supply chain. And that means basically spectroscopy. And if you do spectroscopy, then you need to do black box stuff with it, a little bit like what Nina talked about last week. But so what we do, we were fortunate enough to get this instrument called an aqualog. We begged, borrowed and stole the first half of it, and then the faculty, like with 2 days, you had to have the application in for some equipment funding, and I busted it out because I'd already prepared one for WRI before, and they paid for the rest of it, so now we own this instrument. And it's really cool. So we can record, it's called A-team, so we can record absorbance, transmittance, and then excitation emission matrices, and that's what you see on the right here, which are basically a fingerprint of a wine. And these are 4 different wines here, and even just looking at it at a quick glance, you can see, and they're not massive changes necessarily or differences, but just looking at these, you can see differences there. So these get pre-processed and then get run through some chemometric analysis software using certain algorithms. And Richira has compared a few different ones. And so these are a couple of machine learning ones here. Richira did an analysis of some wines from Australian Cabernet Sauvignon producing regions and did a bit of pre-processing, PLS data reduction, so you don't want all those EM wavelengths and that. You want to reduce that down to a number of components that then you can use for the modelling. That's what Richira did in conjunction with Adam Gilmore from Hariba, so the manufacturer that provides these aqualog instruments. He's a bit of a guru with chemometrics and certainly with the instrument, so he's been very helpful with what we're doing. So support vector machine, that's one type of machine learning, and you can see the accuracy there. Forget about that. Let's go with XGBDA and let's look at 100%, and you don't get any better than that, right? So this is nearly 90 wines and 100% accuracy for each of those regions, even with a small sample set like from Bordeaux. So that's enthused us greatly. So now we're looking at variety, we did, so region then, now we're looking at variety and other regions and trying to expand that. Ruchira is now fusing data sets, so taking that absorbance data, taking the EAM data, smishing these together and doing her prediction modelling on those things. So here we are for the two data blocks brought together, and they're usually complementary data blocks, or should be, to provide complementary information. And so modelling again around geographical origin or indication. And you can see here again, just like before, not quite as good, but geez, one sample out of 221 misclassified is pretty good, I'd say. And if you look at the literature, people aren't really getting that level of accuracy. Yeah, they're in the 90s or whatever, but, and yeah, you know, that might be fit for purpose, but who doesn't want 100%? And we can do the same thing with variety. And then we want to ideally understand what's driving these differences so that the EAMs are picking up compositional things, chemistry, fluorophores, molecules. So can we now start to understand what they are and how these regions differ because of those? So that's what we're doing now. Ruchira is having a look at that. We're also, so that's what's showing here. I won't go through that. And I'm trying, every project I'd almost do now, I'm getting them to do things on the aqualog. So Lyra was doing sensory work on Cabernets and she had a crack at the aqualog. Claire, who's doing things around heterogeneity, has done some things on the aqualog. And the holy grail to me, and I think the battery's just gone flat on this, is predicting wine sensory properties from grapes. How mental would that be? And that's for the path we're heading down now, as well as assessing viticultural treatments or whatever. Smoke taint even, prediction of smoke taint. So there's some really cool things being done, at least in my opinion, with the aqualog. So there's a summary. I'm pretty good on time. So having that background in chemistry, particularly analytical and synthetic organic, has really, you know, underpinned what I do. Basically, they're fundamental aspects of my research. It's really great to apply those to an industry context or applied research. So a total synthesis of a marine sponge molecule. Everyone, you write your literature review, oh yeah, this cures cancer and rah. But then, you know, you do your total synthesis and where are you at the end of it? You've got a molecule that probably doesn't cure cancer. Yeah, you've created some really cool knowledge, and, you know, synthetic chemistry is great, but not just for the purpose of it, which plenty of people do, just to create new methodology and that, and it's super cool, but it's really neat to be able to apply that to an industry that, you know, creates multi-billion dollars worth of economic activity to Australia. I talked about the instruments we use, and particularly around stable isotope dilution assay being important. We're moving into rapid methods, and that's not something that's new for us. Plenty of people, AWRI and the Daniel Cozzolinos of the world, have been doing it for ages, right? But we're taking it in some new directions and using some funky new instrumentation and that. So, you know, making some gains there. And ultimately, a lot of this is cross-disciplinary, and the people I work with and that, or the projects that I work on, aren't just related to chemistry necessarily. So through chemical analysis, though, this is going to link those things and give us the understanding and help grape growers and winemakers ultimately, well, that's the theory, or, you know, that's what we aim for, yeah, to do things to create knowledge that ultimately helps the industry. That's enough of that, I guess. And yeah, I've kind of banged on about the ability to measure things, but also that's not the end of it. It's what you do with that afterwards. So I have a few people to thank. Of course, the people that fund us, the school that employs me, the Waite Research Institute for inviting me, but also being contributors to grants and that we work on. Plenty of people overseas and all my amazing students and DIMI that, you know, that's not me doing that stuff. That's all those guys, right? So, and yeah, I'm super proud of them. And Xinga's got me my 100th paper just a couple of weeks ago. So it's pretty amazing. What a great bunch, hey?
Thank you so much, Dave. For those that are online, if you want to type any questions into the Q&A box, and I'll take questions from the audience, please. Chris?
Great, lovely talk.
Oh, thank you.
That's all. No.
Yeah, cool.
The aqualog stuff, I mean, wine is a changing matrix with time.
Yep.
So
it is.
You know, your data set for N-folds today.
Yep.
If I'm then trying to flog that in 10 years' time.
Yep.
Will you be able to say in 10 years' time that, yeah, this is
In 10 years' time, we will. No, that's the next PhD project. But absolutely, you're right. Even just shipping it across the equator, or things sitting in shipping containers, but particularly this aging thing, whether you can build a model. So Ruchira's done things around vintage, whether they're grouped together or separate, but yeah, whether you can build a model today on Ben Folds, 2020 wine, and it still applies in 10 years' time, it's probably unlikely. But also, this sort of technology, it seems to be, you don't need a lot of wines to build the model, and you don't need a lot of wine, like you need a couple of 100 microliters. So even you could imagine, sampling a little bit of wine during a recorking clinic, or even probably somehow drawing it out with a needle through the cork or closure or whatever, and reanalyzing and rebuilding a model there. So yeah, that's still to be done, but it's a great question. It's an obvious thing to do, yeah.
Any other questions? No? Rachel.
So the obvious question, David, with all this wonderful stuff you can do, is how are you going to, we're going to flood you with, suggestions about what you can apply as alcohol technique to, right? Spirits,
Sure.
food,
Yep.
fermented products,
Yep.
all these things.
That's right.
So you're going to get deluged.
Great.
Deal with that.
I shouldn't have presented it, should I? How am I going to deal with that? I don't know, because I'm trying to wind back the research that I do, because it's not sustainable. So yeah, it's a good question.
So how easy then is it, if we came to you and said we want to collaborate, how easy is it to train people to use those?
Ruchira, what do you think? It took Ruchira, which surprised me, a whole day in the lab to train a new person, but that was, you've got to get the absorbance in the right range and that. So there's a bit of mucking around, but otherwise, what would you say, Ruchira? A day would be enough to train somebody? And if anyone, at the moment, Ruchira is the auto sampler or the person doing it. So one sample at a time, super painful.
Yep.
Ruchira thinks she can do about 24 in 2 hours, so a dozen an hour, or was it 24 in 1 hour? But that's like being a robot, next thing into the sonicator, next 1 hour into the instrument, stir 2 minutes. Like the time for that is nothing. It's 2 minutes to read, and it's really simple sample processing, and that's just a straight dilution and a centro and sonication and that. But yeah, if anyone has $100 grand to help us buy an auto sampler for this, then we'll be able to power through heaps of things, right? Yeah.
It's not such a,
No, I know.
suggestion, because it would have massive applications.
I think so.
Across the whole
I think so too. They're using it to monitor fermentation broths or processes. Yeah, it's amazing. It's so sensitive that I think it has massive, there's still plenty of opportunities to exploit this, absolutely, in other foods, and that, whether it's authenticity or whatever, or just changes, process change. Yeah, for sure. I think, yeah, I'd like to see it being used a lot more. And that's what I said in my application to the faculty when they gave me money, that, yeah, everyone's going to use it. So I'd like that to happen for sure. Once we've done everything we want to, of course.
I might have to bribe you with some.
With an auto sampler?
No. I don't have one of those on me.
Okay, any more questions? Okay, well, thank you everyone for attending, and thank you so much, Dave, for presenting.
No
Very
worries.
interesting talk.
Cool. Thanks. Some more water.